Hello, my name is Dr. Anita Quigley. I'm a Vice Chancellor Senior Research Fellow at RMIT. Today I'm going to be talking about 3D biofabrication and cell culture for tissue repair, tissue engineering and disease modelling. One of the things about tissue culture and disease modelling and some of the areas that people have been moving into recently is tissue culture and biofabrication in 3 dimensions. Now, the primary reason for this is traditionally we've been doing a lot of cell culture for patient cells in 2 dimensions. However, tissues are not 2-dimensional, they are 3-dimensional. So people have been moving into the area of biofabricated scaffolds and 3D tissues for cell culture and for tissue engineering and for modelling of diseases. So cells behave very differently in 3 dimensions. So in 2 dimensions, they have to conform to a flat surface, whereas in 3 dimensions, it's more like the tissue where the cells don't have to conform to this flat surface, and they're in 3 dimensions and they make much better and more contact with the cells around them and the environment around them, so that they're in a much more natural state. People have moved into biofabrication because it actually aids in our researchers to actually grow these cells in a 3D manner, so in a 3D scaffold. This enables us to create complex scaffolds for tissue engineering and disease modelling. So there's a number of different methods, engineering methods involved in biofabrication, and also it's not just engineering, there's a lot of biological involvement as well, as you can imagine, and additive manufacturing techniques. So all of these techniques come together so that basically that's what the field of biofabrication is. It's a merging of all of these fields. So the purpose is to generate synthetic and/or biological scaffolds for tissue engineering and disease modelling, as I said before. And there's lots of new technologies arising in this area that we call it a toolkit. So all of these technologies are being added to the toolkit, things like 3D printing, which I'm going to talk a bit about today. New biologically compatible materials that are constantly being developed. There's new ones coming up all the time. And advances in stem cell technologies and in cell culture also contribute to this, because in a lot of cases when you're doing tissue engineering, you're not just putting in a biomaterial or not just using a biomaterial as a scaffold, but they've got cells incorporated in there as well. So the cells are often a very important part of these scaffolds. So this is really opening the door for a lot of effective treatments for damaged tissues, and it's got big implications in healthcare. For example, the ability to repair tissues, or even for things like using 3D cultures outside of the body using patient cells, so that you can do some drug screening to actually see what drugs are going to work well for that patient. And often the drugs will work differently in 3-dimensional and 2-dimensional cultures depending on what those drugs are. So this can offer a lot to society. There's many instances where our bodies can fail us. We get sick, we have disease, you might have an accident, you might get some trauma, and we're all aging. So many of these ailments, go, you get loss of cellular and tissue integrity, and this is where tissue engineering and biofabrication can step in. This offers a platform to address some of these challenges. So this is really reflected when you actually have a look at the size of the current market, which sits at around $3 to $4 billion in the USA alone. This is the market size for tissue engineering. This is projected to really rise over the next couple of years. So there's a big future in this sort of technology. So, as I mentioned before, tissue engineering can provide patients with a viable options to repair tissue. And one example of that is Apligraf, which is available in the United States. This is a skin graft or an engineered skin graft used to treat diabetic ulcers. It's actually made of a biodegradable polymer mesh, and it actually has some cells on it that lay down an extracellular matrix scaffold. So this company has used biomaterials as well as cells to make a scaffold that can be used for repairing skin. So this is really quite a simple scaffold. In the future, we're envisaging that, it's possible that we could be engineering whole or partial organs. That's where we're hoping the technology is going to move to. And this is going to have, as you can imagine, major implications. You've got things where like partial parts of organs may need repairing, or you might need a whole organ replaced. And there's a lot of people around the world that are actually waiting on the organ transplant waiting lists. And this will hopefully alleviate some of that. We also can use these, as I mentioned before, for disease modelling. And you don't always need to use biomaterials to make 3D scaffolds or 3D tissues outside of the body for disease modelling. Sometimes you get spontaneous organisation of cells as well, and this can be engineered to a certain extent. So these are generally called organoids. There's a lot of talk about organoids around at the moment. And organoids are already helping us to understand some diseases and develop and test new drugs and treatments, and I'll talk a little bit about that later on. So this is an example of how biofabrication can work in the clinical setting. So you might have a patient that comes in with rather a severe skull fracture, and what happens is the patient will then go and get a clinical scan of that fracture, and it's a computer model is made based on the defect. And then biomaterials and stem cells and growth factors are brought together and manufactured to make a tissue replacement construct that gets implanted back into the patient. So this could be a skull, or it could be a piece of bone, or it could be another piece of tissue. So currently there are titanium implants that are being put into bone for people, but the way forward, or the way the scientists are thinking, is actually incorporating some biomaterials, not just metals, as well as stem cells and growth factors to help make something that's a little bit more like natural bone. So the final step of this obviously will be the integration of that scaffold into the patient. That takes a little bit of time. So you have to give, after this is implanted, we hope is going to happen in these sorts of situations is that the body's own cells will move in, and you'll get a meshing between the implant and the tissues around it in the body. So it's a little bit more of a natural implant. So one of the main technologies that's really advanced this field of tissue engineering and 3D modelling is 3D printing or 3D bioprinting. So there's lots of different 3D bioprinters that companies are selling at the moment, and this has really been, there's a lot of companies that have popped up in the last, let's say, 10 years that are selling these systems now, and they're becoming more and more complex and more technical and can do more and more things. An example of a bioprinter that we've got in our laboratories is a GESIM bioscaffolder. Now that can do a number of different techniques. So we can do melt extrusion or electrowriting. So that's what you can actually see here, what this little nozzle is doing down here. It's laying down a very fine line or very fine scaffold of a biodegradable polymer. And we use plastics called thermoplastics for this. So this involves melting the plastic so it becomes into a liquid state so it can be printed, and it will solidify again once it cools down. Now, as you can imagine, this isn't necessarily very good for cells because some of the temperatures that are used here are well above 100 degrees, which, cells are not going to survive that very well. So you essentially have to print the scaffold and then add cells later if you want cells to be incorporated into that scaffold. However, there are other methods that we can use when we actually want to directly print cells. We use hydrogels in general to print cells. So these are things, a hydrogel, they're like jellies essentially. So they're gels that have a very high water content. And these allow us to actually directly print cells or biological factors because we don't need to involve heat. There's no temperature involved in this type of printing. Another method that we can use too is droplet dispensing, and this is a little bit like how your inkjet printer works. We can generate tiny little droplets of liquid that contain individual cells or contain cells and growth factors that can be deposited on the surface. So this machine, for example, brings all of these technologies together into one piece of equipment. so that you can lay down a fine scaffold like this scaffold. You can print in a hydrogel with cells around it, and then you can do some droplet dispensing as well. So you can build up quite a complex scaffold that's designed to accelerate tissue regeneration or tissue growth. This is, we've been calling this convergent biofabrication because it's bringing a lot of different biofabrication techniques together. So as I mentioned before, you can directly print cells using hydrogels. This is a basic printer that we've got in our lab from CellInk, and you can see here, this is just an example of what the printing process looks like. So this is essentially what we call extrusion printing. It's just printing out a hydrogel that contains cells. Once that's printed, we use what we call a photoinitiator that, when exposed to a certain wavelength of light, will cross-link the gel and make it solid. So then we've got a solid structure. And we've used this technique to print muscle constructs. This has been some work done by Catherine, who is our PhD student, who finished up last year, and Robert Kapsa. So I worked with Robert Kapsa at RMIT, and we were previously at the University of Wollongong as well, University of Melbourne. And Catherine is actually an orthopaedic trainee. So she was doing some work on 3D printing muscle constructs that was quite promising, using this hydrogel-based technique where we put muscle cells into the hydrogel and printed them, and I'll talk a bit about those experiments in the next couple of slides. So one of the questions we ask is, can we use a hydrogel-based bioprinting method to create a 3D scaffold, hydrogel scaffold that can be used to form mature muscle? So essentially what we did, this is just a picture of the 3D printed structure. You can actually see all of the cells in the 3D printed structure there. And one of the things with this printing process as well is that, you want to make sure that during the printing process, you're not killing cells. You can see here the green, this is just a little, this area here, you can see the green cells are the live cells and the red cells are the dead cells. Now, you normally will get some dead cells anyway in tissue culture. Because you'll have normal senescence in cells. So these are mouse primary cells, so they had been extracted from a mouse muscle, and what the intention was to bioprint a scaffold with these primary mouse cells and implant them into a rat and bring some vascularization up to the construct as well as some nerve up to the construct, to see if we can get some integration of a vascular system as well as some nerve supply to this construct. So one of the things we did, or Catherine did, to achieve this was go back to an older technology that's been around for a while that a few people at St Vincent's Hospital have published on, is using these arteriovenous loop chambers. We took advantage of the 3D printers that we've got in the laboratory at Biofab. We redesigned this chamber specifically for our purposes. So Catherine redesigned the chamber, put 4 little posts in there so that the construct would lie flat, and also put some access ports in there where we were looking at some electrical activity of the muscle after it was grown. So she took the bioprinted muscle, popped it into the construct, and we implanted it into the groin of an animal. So with this particular model, you redirect the artery, the femoral artery and the femoral vein into a loop so that you're actually getting blood supply to the construct. And you also bring in the femoral nerve. And that's left in the animal for a period of time until it's removed. And hopefully what you get is de novo muscle. You get new muscle forming that's mature. So that is indeed what we saw. We saw after about 14 days, we were seeing areas in the construct where we were seeing quite really what looked like very mature muscle, and which really surprised me. I was happily surprised. We saw striations forming in the muscle that was implanted after 2 weeks, as well as some vascularization coming off the main femoral artery and vein. And you can see the vascularization in green here. You can actually see the blood cells inside the vessels here as well. And we saw, that's just in green here, and we also saw some innervation in red here coming up to where the printed muscle is. So this green area here is the printed muscle, and you can see some little red specks, that's some nerve fibers coming up towards the muscle. So this is really promising. So another area that we're looking at is neural modeling, and mainly 3D. We're moving away from 2D analysis, as I mentioned before. So, you know, as I mentioned, there's fundamental differences in the behavior of cells in 2 dimensions and 3 dimensions, particularly in cell systems where cell-to-cell contact is important. And this is really important in neural systems because we have neural networks. Nerve cells or neural cells in the brain all communicate with each other. And, you know, the brain is a 3-dimensional organ. So, you know, using 3D models outside of the body is a much more accurate way to analyze diseases and analyze how cells are behaving from a patient. So one of the things, one of the other projects we were doing, was engineering 3-dimensional neural cultures. And one of the things that we showed was that when we grew neural cells, these are cells from a rat hippocampus, when we grow neural cells in 3 dimensions and compared them to 2 dimensions, we get activity that's much more similar to the type of bursting activity that you would see in brain. And, you know, it really looked very different to what you see in 2 dimensions. So that just shows that, you know, if you actually want to have a look at brain-like activity in tissue culture, if you're doing drug testing, it's going to be much better doing that sort of thing in 2 dimensions than in 3 dimensions than in 2 dimensions. Now, another exciting area is the area of neural organoids and organoids in general. So, as I mentioned in the previous slide, you can engineer 3D constructs for modeling neural activity using biofabricated scaffolds. You can do this for tissue replacement as well and tissue engineering. But you can also use organoids as 3D models. Particularly if you're doing drug testing or if you're wanting to look at the effects of a particular disease on tissue behavior. The organoids form spontaneously, they're kind of pseudo-engineered. You put them in a situation where they like to form these 3-dimensional structures, and the cells will actually self-organize into structures that resemble the structures that you see in the brain. So Lancaster and Knoblich were the pioneers of this work. It was published in Nature in 2013, so it's been around for about 8 years and it's quite revolutionary. There's been a lot of publications that have come out since then. So the one thing about organoids is that we can achieve a level of complexity that we're not quite there with bioengineering, like directly placing cells cell by cell at the moment. We can't kind of get there at this stage, but hopefully we will one day. So this is why these organoid systems are very useful. We can grow different types of, we can grow different brain regions, and you can see here, this is a picture of the organoids. We actually get very similar structures to what we actually see in the brain. It's really, these organoids are probably the best ex vivo or outside of the body representation of brain-like tissue that we have. As I mentioned before, organoids mimic the structural features and function of organs and tissues. You can see here, this is, I think, a part of the cortex in the brain, and this is what you actually see in an organoid. And they're very similar, have very similar cell types, and these different cell types that arise in the organoid are structured in a similar manner to what you see in the developing brain. So this makes them extremely useful because you can study the effects of disease, and one of the diseases that we're interested in our laboratory is epilepsy. And you can also have a look at the effect of drugs on these organoids. So what we can do with the organoids, we can grow them on multi-electrode arrays, and we can analyze the neural activity, which is what you can see here. Now, when they actually develop neural activity, we say they're functionally active. They're functioning not wholly like a brain, but you're seeing some of the electrical activity that you would see in the brain. So this is just what you can see here. These little spikes represent some neural activity in some organoids that we had been growing. So these organoids now are really being used in a lot, particularly in the neural area of neurological, study a lot of neurological disorders. There's people doing work on Parkinson's disease, Alzheimer's, microcephaly, which is Novlich's group is mainly working on that. And we're interested in epilepsy, and there's a number of groups working on epilepsy as well. So I'm going to leave it there, but in summary, I can say that biofabrication is really revolutionary tissue engineering. And, these methods can be used to create 3D scaffolds, so with or without cells, for the purposes of tissue engineering, and they can be used for the purposes of modeling neural behavior or tissue behavior outside of the body for things like drug testing. So this is a really high growth area at the moment. These techniques are being adopted by many research institutes and companies around the world. And a lot of companies are actually using these biofabrication techniques to make tissue scaffolds that, are on the brink of actually being used in people, particularly overseas. So as I mentioned before, this is really reflected in this, market analysis, where you can see the projected use of tissue engineering products is really on the rise. And these biofabrication techniques of late, particularly 3D bioprinting and 3D printing, has really accelerated this. So in closing, I'd just like to thank everybody from the various labs that I work in and all my collaborators. And I'd like to thank the organiser for the opportunity to speak today. Thank you very much.